So jumping right into it, why do we care about tracking PRRS diversity? So first, PRRS diversity can be used to understand epidemiological links between two different breaks, either breaks in different farms or breaks within the same farm at different times. So that's a possibility of how we can use viral diversity. But also we can try to understand if there are any emergence of new strains of virus or any extinguishing of strains. And that if we do see emergence or strains being emerged or extinguished, we can also try to identify why this might be happening, if there's any trends or any factors that might be influencing the emergence of these viruses. But also, and I think one of the most important things is that our current knowledge says that we do have some cross-protection between different strains of PRRS. So if the viruses are similar enough, we should have some cross-protection against infections. And this is potentially more useful because we can help, this information can help the producers to define a control and elimination program, either being choosing between a commercial vaccination or live virus inoculation. But then again, what defines what is a similar enough virus that would confer this cross protection is still a matter of debate, and I think Igor will get into that a little bit more later. So for our analysis, we're usually using data mostly from the actually all from the MSHMP project, which Kim already explained a little bit. So this is a national program that aims at monitoring swine diseases, and it's very representative of the swine population here in the United States. We currently have 22 states participating in the program with around 38 systems. And other than sharing with us their PRRS status every week, they also voluntarily share with us their sequence data, so their routine monitoring sequence data. So we have data, we have sequence data, or 5 sequence data, from these participants from 1998 to the present, and so far we have over 31,000 sequences. Most of our sequences are type 2 viruses, which are the American type viruses, but we do have some type 1 viruses, which are the European viruses. So with that in mind, with such a rich database, we raised some research questions trying to make those individual monitoring data a little bit, trying to make them a little bit more useful and meaningful for the industry as a whole. So with that, the questions that we raised were, how was PRRS diversity, how did PRRS diversity change over time within that population? Specifically, can we see trends in percent nucleotide divergence from our reference sequences? So when we compare to the historical sequences, how are they changing? Can we see trends in RFLP type sequences frequency or in percent nucleotide divergence from our historical RFLP sequences as well? And lastly, do we see any space-time clustering of PRRS within all of our database? And if we do see space-time clustering, how do sequences inside the clusters compare to sequences outside the clusters? So regarding the first 2 questions, the first thing we did was we used only unique sequences from our database, meaning that every time we had a sequence that appeared in our data set more than once, we only kept their first appearance. And then what we did is we compared that to the reference VR2332, and we did that only for the type 2 sequences, just because it was what we had most in our data set, and it actually doesn't make a lot of sense to compare it with a completely different type. So this reference VR2332 was actually isolated in 1992 and is a parent strain of one of the vaccines that is used in the field. So here what we are plotting is the percent nucleotide difference from each unique sequences to that reference. And we can see here that actually most of our data is from 2005 onwards. But we can see here on the box plots that it sort of seemed like the percent nucleotide difference is increasing a little bit over the years. We can see that the ranges increases a little bit. But we also see that from year to year, we have very different ranges of percent nucleotide diversity. So we have, for example, here in 2007, the range was very narrow, so we didn't have a lot of difference. I mean, the difference per se was was around 15% to the reference, but the range was very narrow. And that was very different, for example, for 2018. We have a very long range. What I also want to highlight here in this graph is that the dots here are the outliers. So we can see that the sequences that are less than 5% different from the reference are actually our outliers. So we don't have a lot of sequences that are too similar to that reference. And that is true even before, for example, here, 2010, which is around the time that the vaccination was starting to get widely used in the field. So moving on to RFLP type sequences, what we did now is we started looking first only at 174s, mainly because we have a lot of RFLP patterns. So if anyone here in the audience doesn't know what RFLP patterns, how they are assigned, is that we have 3 enzymes, and those enzymes, when we have a complete nucleotide match of that enzyme sequence to our R5 sequence, we have a cut, and the amount of cut and the positions of those cuts are what generates the RFLP patterns. So with that, we have a very large possible combinations of the cuts of the 3 enzymes. So we decided to choose just the 174s, mainly because it has been described as one of the most virulent strains or types. It is associated with high mortality in piglets, severe morbidity in sows, and longer time to win negative piglets. So here we're just plotting the frequency of RFLP 174 in our data set, and we can see that it got introduced at around 2003, and it remained at low frequencies until 2014, when we can see that it started increasing in frequency. It reaches peak at in 2015, when 50% of our data set was comprised of 174s, and it appears to be now on a decreasing trend. And in 2018, we have around 20% of our data set being a 174 sequence. So other than that, we did similar to what we did with type 2 sequences. We also took all of the 174s and compared the percent nucleotide difference to a reference. But in this case, we didn't have a very clear reference. So what we did is we used the first 174 identified in our data set, which was again in 2003. So we compared the percent nucleotide divergence from each sequence to the first reported in our data set. And here, the story is a little bit different. We can see that the difference is clearly going down, other than the other graph that we showed before, both in range, but so at first we had around 3 to 15% difference from the reference, and lately we have around 5 to 10% difference. So one thing that I want to highlight here is that RFLP has an important limitation on what we are trying to assess here, which is that it doesn't necessarily directly relate to genetic distance. Just because since we're making cuts and assigning a pattern name for those cuts, any mutation in specific regions that could otherwise be cut or not in the genome, we can have a completely different RFLP pattern. So that relation is not a clear, direct relationship. But still, that difference in range in percent nucleotide identity can possibly be explained by lineage changes, which Igor will discuss a little bit later. So just keep that graph in mind, because he's going to discuss a little bit more on lineage, emergency, and extinguish. And yeah, RFLP patterns 174s are mostly lineage 1, which on his study Igor classifies as lineage 1A. But we do see some lineage 1C and 19 RFLPs 174s too. So just keep that in mind, that this the less diversity that we see here compared to what we saw before is probably due to that lineage change. And lastly, do we see any space-time clustering of PRRS? And how do sequences inside clusters compare to sequences outside clusters? So for that, we are working only with sequences from 2009 onwards, and this is because most of our sequences are from that period of time, just because sequencing got more frequent, more common. We also wanted to avoid repetitive sequences from the same outbreak. So for that, we excluded sequences that were identical and were from the same location and time. But we also excluded identical sequences from the same location up to 30 days from apart from each other. Lastly, we have some clustering of reporting as well. So for that, we decided to divide our analysis into different census, US census regions. So we have here the census regions, and we are analyzing 2 of them separately. And what we did is we're looking for space-time clustering, and for that we used space-time permutation on SAT-scan, accounting for systems. And what that does is imagine a map over here, and we have this cylinder over here, and this cylinder is dynamic in the sense that it is flexible, so we can scan different areas, but it also is dynamic in relation to time. So if we imagine that this is 2009, this is 2010, or this is week 1, this is week 2, so we also can be dynamic in regards to the time scale that we are using as well. And what we are looking for is clusters in which we find a higher number of observed cases that we would expect for that region in that amount of time. And I'm showing here some preliminary results because we are still analyzing this. But for one of the regions, we found 3 clusters so far. And here you can see the number of sequences, farms, and the period that the space-time cluster occurred. So for each cluster, we consider all of the sequences that were not in the cluster for that region, so that were outside the cluster, plus or minus 3 months from the cluster date as control. So just so we can compare the sequences inside the space-time clusters to something else. And what we see here, so these are the phylogenetic trees for the cluster 1, cluster 2, and cluster 3. And what we would expect if we had some, if we had some area factors explaining this transmission, is that we would expect to see something like this, in which the K sequences are the colored ones and the control sequences are the black ones. We would expect to see something like this, that the K sequences are clustered within one clade only. But we can see that that doesn't necessarily happen all of the time. Even for that small cluster, we also have a K sequence that is in another clade. So that relationship doesn't appear to be so obvious. If we compare the mean percent nucleotide identity of sequences within the cluster, we can see that they are usually higher than the mean percent identity of sequences outside the cluster of or control sequences. So they are usually higher among cases than among controls. However, that range can also be so big that they overlap a lot. So even though we do find some associations here, it's not always going to be 100% identical among space-time clusters. We also compared those sequences based on molecular variance according to SNPs. So what that does is we are actually looking at if the genetic variance within the cases is different or not of the genetic variance of the population as a whole. And we did the same thing for the controls, and we didn't see any associations there. And lastly, we also look at RFLP patterns, just because since we are talking about smaller spaces, lineages might not be sensitive enough to describe what is going on, what is the diversity within those clusters. So here we can see that among cases, which are in sequences inside the space-time clusters, we usually have less RFLP diversity than we do among controls. And this is just Shannon diversity index. So the higher the age, the index, the higher the diversity. And again, we can see that within cluster, we usually have much lower diversity than we do within controls. And with that, I'm going to pass to Igor, which will explore a little bit more regarding the diversity of sequences.
So thank you very much. Hopefully you guys will be able to hear me. Let me know if not, then I can change a little bit over here, the way I'm speaking. But with the 10 minutes or so that I have left here, I'm going to talk a little bit regarding genetic and antigenic variability in PRRS and the analysis that I've been running the past year or so here at the University of Minnesota. So we know that PRRS random mutation and recombination occurs. It's a thing. PRRS has been genetically diverse from the origin. We know from the first few times that it got described, we know there are 2 different types. Within those types, there are different clades or clusters that occur. And we believe that perhaps immune selection is a part of, is responsible for the formation of those particular clades. But we also know that those different clades that we observe within PRRS, they elicit different immune response by the host and different cross protection. Talking about PRRS genetic diversity, from the beginning, from the first time that PRRS got described, genetic diversity increased by more or less 20% if we're talking about R5 only. So it's a challenging virus to work with. Again, going back to one of the slides that Mariana presented before, why do we care about tracking PRRS diversity? Personally, one of the things that I like to use this tracking is to continually update our knowledge on circulating PRRS variants. So specific strains or lineages or RFLP types or whatever the label you want to use might occur more frequently in specific periods in time versus in other periods in time. And being able for us to understand why that happens might provide interesting insights for PRRS control. So Mariana discussed a little bit about RFLP, and I'm going to discuss more about this lineage classification. So how does it work? It classifies PRRS type 2, the North American PRRS, into 9 different lineages. This is just one tree that describes this. So those 9 different lineages, they are considered quote-unquote ancestral viral families. It is based broadly on genetic distance and phylogenetic relationship. So instead of being based on specific positions in which specific enzymes cut this particular portion of the virus, it's a form of classifying almost the tree, genetic tree that you construct from the virus. Different lineages have different percent differences to one another depending on which comparison you're making. Roughly, they vary between 10 to 15% between lineages. And again, 9 lineages were described from the beginning, and the expected time that they diverge from one another is prior to 1992. So whatever you see occurring in the United States nowadays is not something that is happening now, or well, things might be further changing now, but the origin happened way a long time ago. So what we did, I'm using the same data set as Mariana was using. So I selected 4,300 sequences from MSHMP from one specific region of the country. I obtained vaccines and the two prototypes, the North American and the European prototype. I also obtained 800 and something sequences that I'm calling quote-unquote anchors. So those were the sequences that the author, this author here, she, used to create this lineage classification back in 2010. So I obtained those anchor sequences in order to be able to classify my or the MSHMP sequences according to those lineages. So I did some data cleaning steps, and in the end I was able to classify my 4,000 and something quote-unquote survivor sequences into lineages. And what I found was this. This is the tree that I found. First things first, the prevalence of these MSHMP sequences according to lineage. 70% of all of the sequences identified in this particular region, I didn't mention before, but I was using data from 2009 to 2017. So during these 8-9 years, 80% of the sequences came from a single, or assigned to a single lineage, lineage 1. Specific lineages just didn't appear on our data set, lineage 2, 3, 4. Other lineages appear with less frequency, but the vast majority of our sequences were from lineage 1. And when we constructed the tree, lineage 1 is this branch that goes from this clade over here that goes from this blue or teal color up to this green part over here. So everything here, this is lineage 1. So there are obvious differences within lineage 1. So this part is different than this red part, which is different than this green part. So we further subdivide lineage 1 into what we are calling, and this terminology is internal. It doesn't relate necessarily to what other people are using outside. But we further subdivided lineage 1 within 5 sublineages, namely 1A, B, C, D, and E. So still, 50% of our sequences were from lineage 1A, 13% from 1B, and 30-something% from 1C. If we are talking about RFLPs, because RFLPs in the end is what people use in the field. So within lineage 1A, 66% were RFLP 174. Within lineage 1C, also 1% were 174. And I highlighted this in green because this is the same RFLP type present in different lineages. Meaning that those viruses, despite having the same RFLP, if we construct a tree, they're located quite far away from one another. In this particular case of 174, they would still be lineage 1A, lineage 1 something. But there are scenarios in which this wouldn't happen. So for example, RFLP 144. So those were 78% of all sequences classified as lineage 1C. And they also were the percentage is correct despite being the same, which is odd. But they were also 78% of all sequences classified as lineage 7. So the point that I'm trying to make here is that when you're using RFLP types, you might be calling different things by the same name. So be aware of that. One thing that we decided to explore also was to try to illustrate how different were our sequences that were classified in different lineages. So this is a table that shows the genetic distance of each lineage and sublineage, the lineage 1A, B, C, and E versus all the others. So, for example, the mean genetic distance within lineage 1A was 2.3%. But if I compare lineage 1A versus lineage 1B, it was 8.3%, and so on and so forth. So genetic distance between lineages was usually between 5 and 13%. Obviously, there is some range there, depending on which comparison you're making. And the genetic distance within lineages tended to be under 5%. This doesn't hold for all instances, but for most of it, it holds. Another thing that the tree that we constructed earlier already made clear, but this also highlights, is there's a huge variability within lineage 1. So if I'm going to call viruses by lineage, and if I'm going to say lineage 1, I might be throwing a lot of different stuff together. We decided to then construct a timeline in which we were plotting the lineage occurrence over time. And what we see was that there were specific lineages that, despite being very common in the beginning of the timeline, they almost went, quote-unquote, extinct over time. Other lineages, namely lineage 1, all variants of lineage 1, they were very rare in the beginning, and then they increased occurrence over time. We then thought to ourselves, how new sublineages form? Can we find evidence for evolution in this data set? So we decided to look for a metric called DNDS. Very shortly, what it does is it compares mutations according to if the mutation was a synonymous mutation or a non-synonymous mutation. And in the benefit of time, I'm not going to go into too many details on that. But what we found was the following. We found specific portions of our 5 genome in PRRS that whenever we run the analysis, we consistently identify high DNDSs, meaning that non-synonymous mutations in those sites were being favored. So we decided to look, okay, what do those sites code for? And we found that those sites were mostly, those sites over here, those 2 peaks over here. Those sites are the hypervariable regions 1 and 2. They flank the principal neutralizing epitope, which, as the name says, is perhaps one of the principal neutralizing epitope of PRRS virus. So how we interpret this? Mutations in those sites are being favored because they change, or they might change, how different antibodies are being able to recognize this principal neutralizing epitope. So the way we are interpreting this data is that this data support the hypothesis that PRRS evolution is at least partially driven by immune processes. And how we think that this happened, if you think that there is a viral population that within a host there is a viral cloud, so not all viruses are the same within a host, and if you have an immune pressure that it's going to create some sort of barrier or bottleneck when transmission occurs, this immune barrier might favor over time the appearance or the selection for viruses that are different from whatever this immunity is protecting against. In this framework, varying degrees of cross-immunity is crucial. You have to have imperfect protection from viruses in order for this to work. So we decided to look on the MSHMP data, and we did see that adoption of vaccination and LVI protocol in this particular region where these sequences came from did not occur in the same time. So we do believe that we can see evidence for varying immunity through time in this particular region. And coincidentally, I'm going to call attention to this pink part over here. So this represents farms that were classified according to the MSHMP status as being positive, stable with field virus inoculation. And you can see that this particular procedure occurred more frequently beginning in 2015 onwards. So I went to those systems and I spoke to them and I asked, hey, what happened in 2015 that you guys decided to use LVI more often? And their response was, quote unquote, because they realized that viruses in that particular time were too different from the vaccine ones. That's why they decided to adopt LVI. Oops. So to finish, starting to wrap up my presentation here, we identified vaccines that belong mostly to lineages that are not the most prevalent in the analyzed data. We saw a high prevalence of lineage 1. We think that this high prevalence of lineage 1 could precisely be because those sequences are better able to evade immune responses elicited by vaccines. I didn't show before, but this tree over here displays, it's kind of small, but as stars, vaccines. And well, when this data was collected, there were no lineage 1 vaccines. However, this is a little bit tricky because it's possible also that the sheer occurrence of lineage 1B and C, which is the green and the red one over here, might have allowed for the emergence of this teal lineage over here, this lineage 1A lineage. So we believe that we are being able to detect this kind of effect of immunity shaping disease occurrence, specifically sublineages occurrence through time. A few limitations. First, this is a convenient sample. The criteria in which different samples were collected, why they were collected, how many animals were collected, it's something that I don't have access to, so it's difficult for me to be able to better refine this, but I expect that the criteria were different according to different samples. I don't have a denominator. I don't know how many samples were actually tested. I just have the positive ones. This only uses R5 data. It's only 4% of the viral genome. What is happening with the other 96% of the viral genome? With this kind of data, I cannot tell you. We are planning on expanding this analysis to other regions of the United States, and we are planning on doing similar analysis in the future using whole genome sequencing. in the past, things were more expensive. Nowadays, this doesn't justify it. It's not, technology changed. We can go in that direction. Some take-home messages. So type 2 PRRS nucleotide divergence from the reference 2332 averaged at around 10 to 15% throughout 20 to 2018. This is Mariana's presentation, what she said. PRRS RFLP type 174 had been an increasing trend, occurrence up to 2014, and appears to be now on a decreasing trend. This corresponds more or less to the same thing that I observed when I classified my sequences as lineage 1A. And sequences within a space-time cluster had a higher mean percent identity and lower RFLP diversity, at least using these kind of parameters. Other indicators did not support the same findings that Mariana had. These highlight the challenges that we have when assessing space-time and genetic relatedness in PRRS transmission. Lineage classification allows us to classify PRRS viruses into broad families, very broad families. The phylogenetic relationship amongst isolates are clearer. However, be aware you might be calling by the same name things that can be different. It seems to me, at least, that it's more intuitive for tracking broad evolutionary trends and making comparisons across space and time. Multiple lineages co-circulate in this US region, and we hypothesized, or the hypothesis that we have, that PRRS evolution is at least partially driven by immunological processes is plausible. The data that we have seen shows that. With that, I would like to acknowledge the veterinarians and the systems that contributed to the data, the funding agencies, the partners, and faculty and collaborators from the University of Minnesota. Thank you very much.